The ubiquitin-proteasome system is a critical cellular process that controls protein degradation. The first step, ubiquitination, is a three-stage process that tags proteins for destruction. The first stage is activation, where a single ubiquitin molecule is activated by an enzyme called E1. The second stage is conjugation, where the activated ubiquitin is passed to an E2 enzyme. The final stage is ligation, where an E3 ligase recognizes and attaches the ubiquitin tag to a target protein. This process is repeated to form a chain of ubiquitin molecules, known as a polyubiquitinated protein. The next step, deubiquitination, is where a group of enzymes called DUBS remove the ubiquitin tag from the protein. The now tagged protein is broken down into smaller peptides and amino acids during proteolysis. The ubiquitin molecules are recycled and used again. Angelman syndrome, a non-functional UBE3A gene, which encodes an E3 ubiquitin ligase, causes this neurodevelopmental disorder. In key brain regions, the maternally inherited gene is the only active copy. The absence of a working UBE3A protein leads to the toxic buildup of its target proteins, like ARC, which disrupts synaptic function. This results in severe developmental delay, intellectual disability, and ataxia. Familial Parkinson's disease. Mutations in the PARC2 gene, which encodes the E3 ubiquitin ligase parkin, cause this familial form of Parkinson's. Parkin clears damaged mitochondria and proteins. When it's not working, dysfunctional mitochondria and protein aggregates, known as Lewy bodies, accumulate. This leads to the progressive death of dopaminergic neurons, causing motor symptoms like tremors and rigidity. Von Hippel-Lindau syndrome is caused by mutations in the VHL gene, a component of an E3 ubiquitin ligase complex. This complex normally degrades the hypoxia-inducible factor, HIF, protein. A defective VHL protein allows HIF to accumulate, driving the transcription of genes that promote uncontrolled cell growth and highly vascularized tumors. Familial Mediterranean fever. Mutations in the MEFV gene cause the pyrin protein to become hyperactive, leading to the uncontrolled activation of the inflammasome, a key part of the immune system. This dysregulation causes recurrent fever and painful inflammation. Nezaloff syndrome is caused by a defect in the A20 gene, which encodes a deubiquitinating enzyme, DUB. A20's function is to remove ubiquitin tags to turn off immune responses. A defective A20 protein leads to a persistent, excessive activation of the NF-kappa B pathway, resulting in uncontrolled immune signaling and severe immunodeficiency. Immunodeficiency with centromeric instability and facial anomalies, ICF. ICF is a complex disorder with mutations in genes like DNMT3B, ZBTB24, and CDCA7, which maintain DNA stability. Their dysfunction disrupts the UPS, leading to chromosomal instability and compromised immune function. Chorea acanthocytosis is linked to the VPS13A gene. A non-functional VPS13A protein disrupts protein trafficking and ubiquitination, causing the accumulation of abnormal proteins. This results in involuntary movements and a characteristic change in the shape of red blood cells. Neurodegenerative diseases. Spinocerebellar ataxia type 17 and Huntington's disease are caused by expanded polyglutamine repeats in the TBP and Huntington proteins. These repeats cause the proteins to misfold and form toxic aggregates that the UPS cannot clear. This accumulation leads to neuronal death, causing symptoms like ataxia, dementia,